أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. In the name of Allah, the most gracious and the most merciful. Today I am delivering a lecture from chapter number 4 of ninth book, Cells and Tissues. For consulting with the lecture, students should follow these general instructions. Now, the first topic which I am going to discuss, that is passage of molecules into and out of the cells. Cell membrane is a semi-permeable membrane because it allows some molecules to pass through and does not allow the other molecules to pass through. Cell membranes maintain equilibrium inside the cell as well as outside the cell by exchanging matter with cell's environment according to needs. Cell membranes do it through the phenomenon of diffusion, facilitated diffusion, osmosis, filtration, active transport, endocytosis, and exocytosis. Actually, these are the processes by which materials can move in and out of the cells. The first process by which the materials can move, that is diffusion. Diffusion is the movement of molecules from the area of higher concentration to the area of lower concentration, that is along concentration gradient. Concentration gradient means from higher concentration to lower concentration. At the end, the molecules reach a state of equilibrium. It means when molecules will move from higher concentration to lower, ultimately the molecules on both sides will become equal. Diffusion is one principal method of movement of substances within the cell as well as across cell membrane. Carbon dioxide, oxygen, glucose can cross cell membrane by diffusion. Gas exchange in gills and lungs occurs by diffusion. Movement of glucose molecules from small intestine lumen into the blood capillaries of villi is also by diffusion. Diffusion is a type of passive transport because a cell does not expend energy when molecules diffuse across its membrane. In this picture, you can see that the molecules are moving from higher concentration to lower concentration. This is diffusion. Second process by which the molecules can move through the cell membrane is facilitated diffusion. When a transport protein moves a substance from higher to lower concentration, the process is called facilitated diffusion. The rate of facilitated diffusion is higher than the simple diffusion. It means molecules by facilitated diffusion moves fastly as compared to simple diffusion. Facilitated diffusion is also a type of passive transport because there is no expenditure of energy in this process. In this picture, you can see that these are the transport proteins, and uh, by the help of these proteins, molecules are moving from higher concentration to lower concentration. So this is facilitated diffusion. The next process by which the molecules can pass through cell membrane is osmosis. Osmosis is the movement of water from a solution of lesser solute concentration to a solution of higher solute concentration across a semi-permeable membrane, or we can say that the movement of water molecules from a region of higher water concentration to lower water concentration. A hypertonic solution has relatively more solute. A hypotonic solution has a relatively less solute, and in isotonic solution has equal concentration of solutes on both. In this picture you can see that this is semi-permeable membrane. This is the solution where water concentration is high and solute concentration is low. In this side, solute concentration is more and water concentration is low. In this case, you can see that water molecule is moving from uh, high water concentration to low water concentration. Now we will discuss about water balance problem in animal cell. When an animal cell such as red blood cell is placed in an isotonic solution, the cell volume remains constant because the rate at which water is entering the cell is equal to the rate at which it is moving out, because the solution is isotonic. The movement of molecules in and out is equal. When a cell is placed in a hypotonic solution, water enters and the cell swells and may rupture like a overfilled balloon. When an animal cell placed in hypertonic solution, it will lose water and will shrink in size. In fresh water, for example, in hypotonic environment, animal cells must have ways for to prevent excessive entry of water. In seawater, for example, hypertonic environment, they must have ways to prevent excessive loss of water. In this picture, you can see that here you can see that the blood cells are present in isotonic solution. The amount of water which is going out is equal to the water going in. In case of hypertonic solution, when red blood cells are in hypertonic solution, the water is moving from inside to outside. As a result, the cells are shrinking. In this picture, where blood cells are present in hypotonic solution, the water is continuously entering into the cell and uh, cells are swelling or they may rupture like a balloon. Next is water balance problems in plant cells. Water balance problems are different for plant cells because of their rigid cell walls. Most plant cells live in hypotonic environment. That is, there is low concentration of solute in extracellular fluids than in the cells. As a result, water tends to move first inside cell and then inside vacuole. When vacuole increases in size, cytoplasm presses firmly against the interior of cell wall, which expands a little. Due to strong cell wall, plant cell does not rupture but instead become rigid. In this condition, the outward pressure on the cell wall exerted by internal water is known as turgor pressure, and the phenomenon is known as turgor. In isotonic environment, the net uptake of water is not enough to make the cell turgid and it is flaccid. In hypertonic environment, a plant cell loses water and cytoplasm shrinks. This shrinking of cytoplasm is called plasmolysis. In this picture, you can see that when plant cell is present in water in hypotonic solution, water is continuously entering into the cell and then into the vacuole. As a result, the cell is becoming turgid. In isotonic solution, when plant cell is in isotonic solution, the water going in is equal to the water going out. It means the cell is neither becoming hard, neither becoming soft. But in this case, where cell is placed in hypertonic solution, here water is moving from inside to outside and the cytoplasm is shrinking. This is called plasmolyzed cell. Now the next topic of the book is osmosis and guard cells. Stomata in leaf epidermis are surrounded by guard cells. During daytime, guard cells are making glucose and so are hypertonic than nearby epidermal cells. Water enters them from other cells and they swell. In this form, they assume a rigid board shape and a pore is created between them. At night, when there is low solute concentration in guard cells, water leaves them and they become flaccid. In this form, both guard cells and rest against one another and the opening is closed. In this picture, you can see that the guard cells have become board shaped. As a result, stomata opens. In this case, the uh, walls of the guard cells they have touched each other. As a result, the stomata has closed. Now we will discuss the next topic, which is applications of knowledge about semi-permeable membrane. Semi-permeable membrane is capable of separating substances. Artificially synthesized semi-permeable membranes are used for the separation of bacteria from viruses because bacteria cannot cross a semi-permeable membrane. In advanced water treatment technologies, membrane-based filtration systems are used. In this process, semi-permeable membranes separate salt from water.
The
Now we
next
will
process
discuss
by
the
which
next
the
topic, which is
materials
applications of
can move through cell
knowledge about
membrane is
semi-permeable membrane.
filtration. Filtration is
Semi-permeable
a
membrane
process
is capable of separating
by which small
substances.
molecules are forced to move across
Artificially
semi-permeable
synthesized
membrane
semi-permeable membranes are
with the
used
help of
for the
hydrostatic pressure
separation
or
of bacteria
blood
from
pressure. It means materials
viruses
are moving
because bacteria
out of
cannot
the
cross a
uh,
semi-permeable
out of the
membrane.
cell membrane by the help of
In
blood
advanced
pressure.
water treatment
For
technologies,
example, in the body of an
membrane-based
animal, blood
filtration
pressure
systems
forces
are
water
used.
and dissolved molecule to move through the
In this
semi-permeable
process,
membranes
semi-permeable
of the
membranes
capillary wall cell.
separate salt from water.
In filtration,
The pressure cannot force
Now we
large
will
molecules such
discuss
as
the next
proteins
topic, which is
to pass
applications
through the
of
membrane pores.
knowledge about semi-permeable membrane.
In this picture, you can see that
Semi-permeable
this is
membrane is
the
capable of
wall of the blood
separating
capillary.
substances.
In this case, the pressure of
Artificially
the blood is
synthesized
greater
semi-permeable
inside
membranes
the
are used
blood
for the
capillary. So,
separation of
due to high
bacteria
pressure,
from
small molecules
viruses because
are
bacteria
moving
cannot
from
cross a
inside the
semi-permeable membrane.
blood capillary to the outside of the
In advanced
blood
water
capillary.
treatment technologies, membrane-based
The next
filtration
process is
systems
active
are
transport.
used.
Active transport is the
In this
movement
process,
of molecules
semi-permeable membranes
from an
separate
area of
salt
lower
from water.
concentration to the area of higher concentration.
Now we will
This
discuss
movement
the
against
next
the
topic, which is
concentration gradient
applications
requires
of
energy
knowledge about
in the form of
semi-permeable
ATP.
membrane.
In
Semi-permeable
this
membrane
process,
is
carrier
capable
proteins
of
of cell
separating
membrane
substances.
use energy to move the molecules against
Artificially
the
synthesized
concentration
semi-permeable
gradient,
membranes
that is
are
from low
used
concentration to
for the
high
separation of
concentration.
bacteria from
For example,
viruses
the membranes
because
of nerve
bacteria
cells
cannot cross a
have carrier
semi-permeable
proteins in the
membrane.
form of sodium potassium pump.
In advanced water treatment technologies,
In a resting nerve
membrane-based
cell,
filtration
this
systems
pump
are used.
sends energy ATP to maintain higher
In this process,
concentration of potassium
semi-permeable membranes
and lower
separate
concentration of
salt
sodium
from
inside the cell.
water.
For this purpose,
Now
the
we
pump
will
actively
discuss the
moves
next
sodium
topic,
to the
which is
outside of the cell
applications of
where they are
knowledge about
already in higher
semi-permeable
concentration.
membrane. Semi-permeable membrane is
Similarly, this
capable of
pump moves
separating
potassium from
substances.
outside to inside and inside the
Artificially
cell
synthesized
where they are in
semi-permeable
higher
membranes are
concentration.
used for the
In
separation
this
of
picture, you
bacteria
can see
from
that potassium
viruses because
ions are
bacteria
greater
cannot
outside
cross a
and sodium
semi-permeable
are greater.
membrane.
Sorry,
In advanced water
the
treatment
potassium ion are
technologies, membrane-based
greater
filtration
inside
systems
and
are
sodium are
used.
greater outside. These are the sodium
In this
potassium
process,
pumps
semi-permeable
which can move the
membranes
sodium ions
separate salt
and
from
potassium ions
water.
simultaneously.
Now
The
we
next
will
process
discuss the next
by
topic,
which the
which is
molecules can
applications of
move through the
knowledge
cell
about
membrane is
semi-permeable membrane.
endocytosis. It is a process of
Semi-permeable
cellular
membrane is
ingestion
capable of
of
separating
bulky materials
substances.
by the infolding of cell membrane in the
Artificially
form of
synthesized
vacuole.
semi-permeable membranes are
Endocytosis
used
is of two
for
types,
the
that is
separation of bacteria
phagocytosis
from
and
viruses
pinocytosis.
because bacteria
In
cannot cross a
phagocytosis,
semi-permeable
cell takes in
membrane.
solid material, that is
In
cell
advanced water
eating.
treatment
In
technologies,
pinocytosis, cell take in
membrane-based filtration
liquid
systems
in the
are
form of
used.
droplets. That process is called cell
In this
drinking
process,
or
semi-permeable
pinocytosis.
membranes
In
separate
this picture,
salt
you
from
can see that
water.
bulky molecules are moving inside the
Now
cell
we will
by
discuss the
infolding of
next topic,
membrane.
which is applications
Now
of
the molecules have
knowledge
entered
about
into the
semi-permeable membrane.
membrane and they have
Semi-permeable membrane is
separated from cell
capable of
membrane in the
separating
form of vacuole.
substances.
This entrance of molecules by infolding of
Artificially
membrane is called
synthesized semi-permeable
endocytosis.
membranes are used for the separation of
Now the
bacteria
next
from
process, the last
viruses
process is
because bacteria
exocytosis. It is a
cannot cross a
process through which
semi-permeable membrane.
bulky material is exposed by
In
bursting
advanced
of
water
membrane.
treatment
This
technologies,
process adds new
membrane-based
membrane
filtration
which
systems
replace the part of
are
cell
used.
membrane lost during
In
endocytosis.
this process, semi-permeable
In this picture,
membranes
you can see
separate salt
exocytosis.
from
The cell
water.
is capturing materials from inside the cell
Now we
and they are
will
making
discuss the
vesicles or
next
vacuole.
topic,
When
which
this
is
vacuole
applications of
will go near to cell
knowledge about
membrane, it bursts,
semi-permeable membrane.
releasing proteins or waste
Semi-permeable
materials
membrane
out of the
is
cell.
capable of
And
separating
the
substances.
cell membrane of the vacuole fuses with the cell
Artificially
membrane.
synthesized
This
semi-permeable
process is called
membranes are used
exocytosis.
for the separation of bacteria from
So I have
viruses.
completed my lecture,
Because
and at
bacteria
last
cannot cross
of
a
the lecture I am
semi-permeable
giving the
membrane.
students
In
assignment
advanced water
number 1.
treatment
You have
technologies,
to complete it and you
membrane-based
have to
filtration
submit it on
systems are
WhatsApp
used.
page. The
In this
first
process,
question of assignment
semi-permeable
is what is
membranes
diffusion? Give
separate
its
salt from
importance.
water.
What is Turgor pressure and plasmolysis?
Now we will
Third
discuss the
question
next
is how
topic,
does
which is
stomata open
applications
and
of
close?
knowledge about semi-permeable
Next
membrane.
question, what are the application of
Semi-permeable membrane
semi-permeable
is capable of
membrane?
separating substances.
Next question is what is the function of Turgor?
Artificially synthesized
Next
semi-permeable
is what is
membranes are used
endocytosis? Give its
for the separation of
types.
bacteria
I have now
from
completed my
viruses.
lecture and
Because
also have
bacteria
given
cannot cross
student
a
assignment which they
semi-permeable
can complete after
membrane.
consulting the lecture. So
In
thank you very
advanced
much
water
for
treatment
uh,
technologies,
listening this lecture.
membrane-based filtration